What if the first organ to age isn't your brain, or your heart, or your muscles, or your immune system? What if the chronic inflammation we often associate with aging isn't simply a consequence of getting older, but one of the forces driving it? Can a simple blood test tell us which of our organs is aging fastest? Could your immune system be 10 years older than the rest of your body? And perhaps the biggest question of all, can we measure these changes early enough to actually intervene? Welcome back to Decoding Longevity, the podcast where we explore the science that's redefining how we understand, measure, and ultimately slow human aging. I'm your host, Dr. Raghav Segal, joined, as always, with my co host, Dr. Max Wachakis.
Hi everyone. In our last episode, we spoke about measuring the pace of biological aging and how aging clocks are changing medicine. Today, we're exploring another fascinating dimension of biological age, the aging of the immune system. Our guest is Dr. David Furman, director of the Stanford Thousand Immunomes Project, director of the AI Bioinformatics platform at the Buck Institute, entrepreneur, and one of the pioneers behind the iAge inflammatory clock organ age clocks and leading iAge intrinsic capacity project. David's work combined immunology, artificial intelligence, systems biology, and multi-omics to answer one of the biggest questions in medicine. David, welcome to Decoding Longevity.
Thank you so much. Thank you for having me, Maxu and Dr. Raghav Segal. How are you?
Yeah, it's so good to have you here, David. We've talked so many times, and it's amazing to finally have you on the podcast. So I kind of want to start at a very high level here, which is many people think aging kind of starts in our DNA or our mitochondria, but you've spent a lot of your time sort of thinking about the immune system and how that might actually sit at the center of aging itself. Could you tell us a little more about that? Why is the immune system so critical?
Yeah. So this started in the early 2000s when I was doing my PhD in cancer immunotherapy. So we're trying to boost the immune system to fight cancer. And I realized by reading a lot of literature that at that time there was this general idea that inflammation may drive hallmarks of aging and may drive other aspects of aging, including cancer development and diseases. So that was pretty shocking to me that inflammation and the immune system were involved in aspects of aging. But it wasn't until I moved to Stanford and we set up the whole Thousand Immunomes project that we started realizing which factors were contributing the most. But this is very new, right? We have only 2.5 decades realization that the immune system is not only protecting us from infectious disease, right? It's not only causing autoimmune disease if it goes awry, but it actually accelerates aging. That was a pretty substantial realization.
Yeah, it's very fascinating because almost every disease that we actually associate with aging has an inflammatory component, as you mentioned. Rather than being just a consequence, inflammation may actually be one of the drivers, as we have seen time and time again. And so much that we have created this term called inflamma aging, which has become one of the defining concepts in longevity science and one of the hallmarks as well. But is often misunderstood. What is actually inflammatory aging, if you could explain to our listeners, is and why does our immune system become chronically activated as we get older?
Right. So inflammaging is simply a term that the Claudio Franceschi in the University of Bologna coined to capture the systemic and chronic inflammation that happens with aging. Usually, when we think about inflammation, we think about the infection. We think about red, swollen, warm tissue. That's acute inflammation. And there are very well established markers in the clinic. You can measure your interleukins and your HSCRP, C-reactive protein, and all of those. In the case of age related, this is systemic, it's not local. it's chronic, and it's low grade. It's not so high when you measure the levels in the blood, and there's been no well-established markers in the clinic. And so, why have or the question that you pose is how inflammation starts. I think it starts very, very early, actually in the womb, even before you're born, right? An inflamed, expectant mom that's maybe smoking or obese or overweight will carry some signals and expose that growing fetus to high levels of inflammation. So, our theory that this starts in the womb has largely an epigenetic component, right? You're changing epigenomes while that baby is growing. But most importantly, the immune system acts as a sensory system. So anything that you do to your body, your immune system will record and will react upon. And that reaction is inflammation, right? So if you're constantly bombarding your immune system with triggers, and that could be poor sleep, it could be your croissant that you ate this morning, right? That's highly inflammatory, right? You're going to have inflammation, yeah, inflammaging as we age.
Yeah. And one thing I actually really appreciate here, David, is you don't really see inflammation as simply good or bad. It's you see it as kind of essential for survival. The problem is kind of when it's properly not switched off, right? And that has kind of led to one of your biggest contributions, which is iAge. For listeners who are unfamiliar with it, what exactly is iAge and how is an inflammatory clock sort of different from some of the methylation clocks and other biological age measures? And just
Yeah.
FYI, for our listeners, iAge has become really popular. It's being used as one of the endpoints in the XPRIZE health span. And David, maybe you can share a little more about that as well.
Correct. So the first question, the lack of biomarkers of inflammation led us to just create this big endeavor, which is the 1,000 Immunomes Project. I'm sure we're going to talk about that later. But essentially, it's 1,000 people that came to the clinic for a flu shot. And we leveraged this opportunity to get the multiomics done even before it was called multiomics. We're doing proteomics and cell composition and gene expression and all of that. And you're certainly right. I want to touch upon the idea that inflammation is not bad. It's a evolutionary conserved mechanism to protect us. That's the good fire, right? And then there's the slow burn, right, which is the bad aspect. A lot of research, especially from Charles Serhan at Harvard, demonstrated that there is this switch off, right, mechanism of anti-inflammatory molecules, that when you're triggering your immune system constantly with your poor lifestyle, you can no longer recover that immune system health because you don't have enough anti-inflammatory molecules. But that aside, you mentioned the IH. We took a very unbiased approach to look at the immune system as we age. And from all of the data that we collected, genes and proteins and cell composition and metabolites, the most predictive, as far as calendar age, as well as multimorbidity and mortality, was proteomics. And then we decided, okay, how do we treat this very noisy and redundant data? And we decided to apply a nonlinear model, which is a deep neural network, to create the inflammatory age metric. And then we went ahead and validated that in multiple cohorts. Turns out that now it's been used all over the place, and I'm very, very happy that the XPRIZE Health Span competition selected one of my companies that's commercializing the inflammatory age as the partner to measure immune health.
Yeah, that is super interesting. And what I think it's really great is that, like the epigenome, for example, the inflammation is potentially modifiable as well. So it's different than chronological age that, it's just passing by, but we can do something about it. So you mentioned a little bit about your how it came about, the Stanford 1,000 Immunomes Project with the people coming for the flu vaccine. But could you explain a little bit more about how this developed into this project? And the second part is, since this is potentially modifiable, what are the things that you seem to have the most success so far to actually, have an effect on this inflammaging?
So we defined those biomarkers, we identified the most predictive ones, and we brought them from the academia to the industry, right, with very high standards and CLIA and all of that, which is a clinical test. Now, the triggers, as we know, are exposome factors. And the most predictive proteins are only five, and remarkably, two of them increase with aging, and three of them actually decrease. So, lower levels of these proteins increase your inflammaging overall, which means that these proteins are protective. And some of them are very key aspect of the immune system, like gamma interferon, that helps us against cancer and the antiviral response. And there's also another protein called TRAIL that sensitizes cells to undergo apoptosis, especially cell senescence. So if you don't have enough TRAIL, you're going to have an accumulation of cell senescence in the body. And then there's these two kind of not so friends proteins, CXCL9, that causes accelerated aging of the heart and vasculature, and eotaxin 1 that can go to the brain and disrupt function and accelerate cognitive deterioration. So that's kind of overall the proteins. And you mentioned interventions, right? The interventions, because this is modifiable, we believe that changing your lifestyle and reducing the triggers, it's going to have a huge impact in your overall inflammation. We've tested that. There's a couple of published interventional trials. One of them used beta glucan to increase microbiome diversity and decrease inflammation with some interesting polyphenols that are found in oats, for example, that are called avenantramide, and these decreased inflammatory age by about 3 years in only 2 weeks of consumption. So it's very malleable. The immune system, the beauty of this is that the immune system is so malleable that you can change the course of those trajectories quite easily.
That's amazing. David, I do want to shift gears here a little bit and actually start by congratulating you on the launch of OrganAge with Superpower, which is amazing research to move this sort of work from lab to sort of the clinic. For years, we have talked about this idea of biological age not being a single number, but, and in my own work, we do this, but OrganAge sort of challenges that idea. And instead, it kind of suggests that different organs can age at different rates. So tell us more about this inspiration around the shift in this thinking and why a single biological age is not enough anymore.
Yeah, that's a great question, Raghav. Um, as you know, many in the field have demonstrated that organs age at different speeds. And the speed of your older organ is really something personal. It could be different in your case than in my case than in Maxu's case, right? And so, because this link between inflammation and accumulation of damage in the tissues and organs was so clear to me, and the fact that all these papers are coming proposing with the idea that organs age at different rates, we set out to democratize and understand how we could easily measure organ age without having an epigenetic test done, without having a proteomics complicated, like thousands of proteins, simply using biochemistry labs that you get in your yearly checkup. And so we leveraged the UK Biobank with 500,000 people. And we took all these labs that include your LDL and your creatinine, your cholesterol, and all of those. And we apply machine learning models to predict mortality, cause specific mortality. That's really what our organ age is all about. We're predicting cause-specific mortality that falls into what we call the ICD-10 chapters, right? The International Classification of Diseases. It could be cardiovascular diseases, it could be musculoskeletal, metabolic, your respiratory, and so on. And then we transform that risk score into an age range so that you can interpret, so that you can see what is your weakest organ. And so if you had a high risk of death, right, due to cardiovascular disease issues, we can say this is your cardiovascular disease risk, and therefore you have accelerated cardiovascular aging. That was validated in the health and retirement cohort with more than 10,000 people. So and we're currently offering this as a B2B and going direct to consumer in October, so very soon.
Hmm. That is quite amazing. So we're talking about your organ's age. We know that Raghav has worked in systems age. His work is based on epigenome. Yours is work is based on biomarkers that you can get from biochemistry, as you mentioned. How is that different or comparable? And how do you see, those predictions being, how much they compare?
Mm-hmm.
Are they as accurate as each other? Did you see any difference by using simply the biomarkers or comparing with epigenetic markers?
Yeah.
And what was the one that surprised you the most? Because when you think about the heart, yes, we have biomarkers that are quite, related to the heart, like, as you said, cholesterol. But when you talk about brain, it's, it becomes a little bit more blurred. So how was there
Well,
something
yeah. No, right. This is an interesting question, and the way we framed the study, it was a completely unbiased. We didn't have, cardiovascular specific markers in mind, right? We simply took all of the biomarkers and in an unbiased way predicted mortality in that maps ICD-10 chapters, right? And so it's super interesting to see that molecules like cystatin C, that's produced by kidney damage, right? When you have kidney damage, you have increased cystatin C. And a doctor will say, yes, cystatin C, it's a kidney protein. Well, it turns out it's the most significant predictor across systems, including the cardiovascular system, even more predictive than the LDL. So this is not a reductionist approach. This is a systems medicine approach. We're not looking at textbook biomarkers. We're looking at an unbiased machine learning approach and let the algorithm choose what's most important. You mentioned kind of a comparison against epigenetic clocks and those system-specific ones, and I can extend that to proteomics. Right, there's been quite a few papers where proteomics has been used to predict organ aging. I would say when you compare side by side metrics like area under the curve or C index, C indices to predict accuracy. This is accuracy. That's one big piece of the puzzle, right? We are doing much better than any of the existing systems and organ clocks. And I think the reason is one, we're training on 500,000 people, not 5 or 7,000 or 10,000, right? And two, the biomarker is much closer to the phenotype than epigenetic biomarkers. They're farther away. And then proteomics is very noisy, it's not doing as well. In terms of accuracy, there's another piece which is precision. Precision is test retest. And some of these, epigenetic clocks and the proteomics clocks are based on technologies that are rather new, relatively novel, and a little bit noisy and still developing. So you've probably seen the Matt Caberlain podcast where he sent samples all over some labs and he got very, very different epigenetic ages, spanning 25 years. So I think there's still a lot to learn from those. I love them for research. I believe that having a lab based organ specific clock gives you a much higher precision and much higher accuracy for the reasons I already mentioned.
Yeah, absolutely agree. I do want to again shift gears a little more here to your
Let's do
work
it.
around IC specifically, David, which has been pretty amazing. WHO has sort of increasingly sort of shifted this conversation from lifespan to intrinsic capacity. And your work has recently moved in that direction as well. You have built sort of the DNAM predictor, DNA methylation predictor of intrinsic capacity. More recently, sort of been involved in the ARPA-H project as well. How do you sort of see the future of biomarkers evolving? Is it more like biomarkers like iAge that help us monitor, or are there biomarkers like intrinsic capacity that help us preserve
Mm.
and look at disability long before it develops?
Function. Yeah, function. 100%. I love this question. And to me, the answer is not so much if we want to track one thing over the other thing, but rather, can we have trajectories, mapping rates of change? And of course, second generation clocks have been designed to predict mortality and to predict diseases, which I think is much more informative than predicting simply calendar age. So I think the whole field is moving towards that direction of integrating multi-omics, like third generation clocks, and then being able to predict trajectories, right? Like the Dunedin pace of aging or the intrinsic capacity age that we have now under the review in one of the Nature journals. And you mentioned intrinsic capacity. I think the concept of intrinsic capacity is being very appealing for the aging community and for the longevity researchers. And why is that? Because for many decades, aging researchers like me have been pushing the idea that aging should be considered a disease, but that's never going to happen. Let's state things as they are, right? It causes a lot of social and political uncomfort, and so I think, and it shouldn't be, maybe, right? I don't know. From a purely scientific standpoint, I love the idea. From a more social and ethical angle, I think it's debatable. But in 2022, as you mentioned, Ragav, the WHO for the first time said no, we're going to consider declining function that is associated with age as a disease. And they put it in the ICD 11. Now, the new ICD code has decreasing function as a disease, which is critical because you can now develop, diagnostic tests and interventions tackling declining tissue capacity and bring it to the FDA and get it approved. And it's going to be free for everyone, right? So it's a major change in the way we study aging, and that's why most aging centers now are focusing, shifting focus towards understanding intrinsic capacity, including these big efforts from the government, like the ARPA-H, right, funded research.
Let me just take a step back here. So we talk a lot about intrinsic capacity, and it's very common in the research field, but just for the sake of our listeners that may not be on the research, how do you define intrinsic capacity? What does that mean? Taking a piggyback on the ARPA-H project that we mentioned, that it's major funding for discoveries. What is it that it is getting it right? And do you think, like, where is the greatest opportunity to accelerate longevity research over the next decade, potentially with this project or other projects as well?
Yeah, that's a great call. Thank you, Maxu. So, intrinsic capacity is simply the capacities that an individual has from both physical and mental, and this represents, in a way, well-being. So think of your cognitive and mental health, right? Your psychological capacities, your nutritional, or your ability to be independent, right? So that can be measured by locomotion. and so hearing capacity and vision, acuity, stuff like that. So all of these different tests that are 5 domains of human function have been measured in the clinic in gerontology fields for many decades, but they haven't been explored, and we don't know much about the biology that drives those changes over time, right? So now for the first time something's happening really, really cool, which is gerontology is bringing their tools, the doctor that looks at older people, right? And gerosciences, which is us, are bringing other tools, and now there's an integration of these two major fields. um, and we're discovering all these biomarkers of intrinsic capacity. We're developing interventions to slow down declining intrinsic capacity, and these are precursors of diseases. So it's a really beautiful framework. And you mentioned, yeah, ARPA-H funded research. The Prosper program was launched about a year ago, and we applied. We got the funding, and this is in collaboration with Stanford, and I'm the PI of the Buck Institute. And the idea here is to restore, to be able to, one, identify what are the biomarkers that are more predictive of intrinsic capacity and mortality 20 years out, right? And then develop ways to restore those. So it's a very comprehensive program, very, very ambitious. We want to bring all these things through the FDA in less than 5 years. And I think that's where the whole field is headed. You ask me, where is the longevity field headed? I think to restore function, to be able to identify those that are aging fastest in their organs and their intrinsic capacity decline, and restore function to extend health span.
It's an amazing framing, David. Now, before I let you go, we typically ask two questions from our guests. One is, if everything that you're doing is successful, whether sort of building all of these novel biomarkers, how do you see sort of longevity science dramatically changing in the next 5 years? And the second question I have is, who would be one scientist that you would like us to invite to Decoding Longevity podcast, who you'd like to hear? I know we're putting you on the spot, but we do this with all our guests, I promise.
Well, those are major questions, but the first one is I want to answer by kind of telling you and Maxu what we're headed in terms of my own research, right? So we are developing all these biomarkers. We're understanding more biology. We're putting them out there with partners and with consumers and clinics, and they're being used. I think the major hurdle for large scale adoption at the kind of globe type of level is access, is cost, right? And is the just simply the ability to put the word out there, and people don't know about these things. So what you guys are doing, it's amazing. I congratulate you, and I love what you're doing, and we should do more of these things, but also develop simple biomarkers that you don't have to have your whole proteome analyzed, right? With a few biomarkers, even like the digital biomarkers. There's like, I don't know, 5 billion smartphones out there. While there are digital biomarkers with a photo, the healthy selfie that I call that, you can predict inflammation, right, in the body. There's an error, right? It's not perfect. It's about 60 to 60%, 65% accuracy. But as the field is going towards that direction, my end goal, Raghav, you're asking me what's your big end goal and big plans here, is to democratize longevity tools and put these things in the hands of billions. So that I hope that answers the question, the first question. On the second question, let's see. I'd love to bring. I don't know if you've brought George Church. That would be a terrific guest. I really like George, and for as much as I've known him for a while, he doesn't show up in person. So this is a perfect setting for you guys to interview him.
Yeah. Well, maybe we're gonna be lucky at the biomarkers of aging this year. He will be giving a talk over there.
Mhm.
And here I am promoting the conference for those that haven't
Great.
sign up. Yeah, it's a great conference. I remember seeing you talking there, David, last year, and showing this clock with the picture and just the image. And I remember you showing the picture of you on the airplane and showing how you had a low biological age because you're
That's
flying and
right.
the jet lag and all of it.
That's right. Yeah.
So, super interesting. Thank you so much for coming on the podcast and sharing a bit all of your projects and what intrinsic capacity and everything that is going on and how you're really trying to simplify and really democratize aging research for everyone. And so
Yeah.
thank you so much for your time.
Thank you both. Thank you for having me.
And thank you, David. And to our listeners, we'll link David's work on the iAge clock, the organ age clock, and intrinsic capacity in our show notes. If you enjoyed today's episode, don't forget to follow, share, and leave us a review. Until next time, keep to Decoding Longevity. Thank you.